Our final discussion of novelty is going to involve the origin of the vertebrates. Now, in all of the other cases, there's not ever going to be one single feature that we can say this is 100% the reason why this group is so successful. However, oftentimes there's one that plays maybe the most major role, maybe even explains a majority of that group's success. With vertebrates, as the name implies, the vertebra is important, but there are many, many other features that have been added on, that have been built on in various vertebrate groups that have also been responsible for much of those groups' success. So I just want to reemphasize this point. And so for vertebrates, we will talk about the vertebral column, particularly, well, the more general term, the notochord. But realize that for all groups, we're really building and building on new novelties to make more and more success. And so all organisms, particularly the very advanced ones, can be looked at as a succession of key innovations, that each one makes them successful and diverse, and then eventually there's a new key innovation that makes a subgroup of that even more successful and more diverse, and so on and so forth throughout the history of life on Earth. So we're going to begin with the vertebral column, or more generally, the notochord. The notochord is, early on in the history of the vertebrates, a structure that provides a rigid support along the anterior to posterior axis. In addition to support, it also provides protection for some of the tissue around it. So initially, the notochord is right along the nerve cord, and although it doesn't surround it like our modern vertebral column does, it does provide both rigid support so it doesn't get bent and damaged that way, and also just mechanical protection because it is right next to it and can be hit or bumped without causing damage to the nerve cord, which is below it in the earliest vertebrates. So it is a precursor to the vertebral column, and in addition, it also serves as a critical center for organization. It is an organizer. So remember that term back to the Spearman-Mangold experiments. An organizer is a group of tissue that sends out a signal that then basically tells the cells around it what they're going to become relative to their position in relationship to that organizer. Okay, and so it serves as a critical organizer for the central nervous system. We'll look at that role and the outcome of that role in some of the higher vertebrates. So here's a very diagrammatic representation of a early embryo, but this shows the main features. The notochord in green here, which is just ventral to the nerve cord in red and provides some protection for it, and eventually will surround it in the higher vertebrates and provide a great deal of protection. We have other features also that are maybe not as critically important, but also features of vertebrates and were key innovations and were important for earlier vertebrates, but many of them have since evolved beyond needing these structures. So in orange here, we have gill pouches. Again, in fish and some other groups like amphibians, those do develop into gills. In the higher vertebrates like the mammals, the reptiles, the birds, these still provide an important feature. They provide support and structure for the pharynx, but they're no longer serving their primary purpose as support for the gills. And then finally, a postanal tail, which again is important in some groups and less so in others. So the notochord was originally developed in vertebrates by recruitment of a gene called brachyury. So that's a good gene to know. You should know it. It is the signal, essentially, the initiator for notochord development. Now, brachyury is not a brand new gene building a new thing. It's an old gene that's used in protostomes, in echinoderms, in hemichordates. They do have brachyury also, but it doesn't play a critical role. So brachyury is co-opted, and we end up with this proto-vertebrate, essentially. Right? So this is a chordate. This is actually a urochordate larva. And so we can see what these early primitive vertebrates, prior to them being true vertebrates, but as chordates, would have looked like. In green, we have the notochord, that support structure. In blue, we have the dorsal-ventral nerve cord. And a lot of people get those two conflated. They mix them up or think they're the same thing, but they aren't. The notochord is supportive tissue, muscle, bone, eventually will develop into the vertebrates in these higher groups. And the nerve cord, as the name implies, is nervous tissue, right? So sending nerve pulses, nerve signals. And those are two very different functions for those two structures. Okay, so let's just review a little bit of these organisms that we're going to be looking at, and we'll spend most of our time looking at the higher vertebrates. The urochordates, sometimes called sea squirts or tunicates, as adults do not look very chordate-like. However, they are true chordates, and as larvae, they have all these chordate features. They have a notochord, a nerve cord, they have gills and gill arches, and they have a postanal tail that they use for swimming. Sometimes these are called tadpole larvae. They're not closely related to amphibians. They're very distant relatives of them, but they have the same basic body form and swimming mechanism. What happens is these tadpoles will swim around until they find a likely substrate. They will then attach to that substrate and go through a metamorphosis where the tail and many of the other vertebrate features basically dissolve, and they end up as these, they're very clearly animals. They have nerves and some musculation, but they don't have a clear head. They don't have a centralized nervous system. It's more diffuse at that point. So a little bit of a extreme lifestyle for a chordate. Don't worry about the co-option of genes. That's just something we already just talked about. Brachyuria recruits all sorts of new target genes, and that's how it becomes an organizer. That's all we're doing. And of course, the vertebrates are the most successful group. Incredibly diverse ecologically, large, dynamic, very speciose, many species, especially for large animals. It's incredible the number of species that we have. Almost all large niches are filled by vertebrates. Okay, so an incredibly successful group. So what makes them so successful? Well, we've already talked about the notochord, which evolves and becomes the vertebral column. So that certainly is one thing, and the innovation for which the group was named. But there are many others. So let's add to our list. So when I say what's key innovation for vertebrates, you really should be thinking of a list. Number one, the notochord, which becomes vertebral column, right? Number two, which is maybe a little less obvious, are these things that we're going to call cranioplacoids and neural crest cells. Now, these are organized and patterned by Hox genes, and also their position kind of generally is patterned by the notochord. So the notochord serves as an organizer for here's where these placoids are going to develop, and then they're developed differently by Hox genes that then pattern them for these brand new things. And so we have a relatively simple, pretty homonymous, remember that term? The front segments are much the same as the back, with a little bit of differentiation, but not much. But it's not long until we get complete differentiation where things growing in the front are very different in the middle and in the back and all the way to the tail. And so we get wide number of different body regions, and the Hox genes are responsible for this. But the ground plan, the raw clay, so to speak, the raw cells that will become these differentiated regions are originally set up and patterned by that nerve cord. But in the head, they have very different outcomes. So these cranioplacoids are little bundles of tissue that eventually become nerves. And these become the very concentrated nerves that make up the complex sensory system in vertebrates. So when I say cranioplacoids, you should be thinking eyes, ears, mouth, and all the taste buds that come along with that, all the nose and the olfactory things. Those are all arising from cranioplacoids, these concentrated nerve cells that then take on very different functions. And in some organisms, we even have kind of highly specialized ones. So there are some in the nose of sharks that sense electrical fields. So basically have kind of like a sixth sense, but they're all from these cranioplacoids. Okay? And so they are regions of thickened ectoderm. Remember, the nerve cell is ectodermal in origin, and they have various positions in the head that can correspond to these more complex sensory lobes and then connect in and send those senses into the brain. Some of these are even tactile, right? So they have chemoreception or tactile reception also. So eyes, ears, nose, mouth, all of this, the fine senses that we have in our face or that cats have, like with whiskers. Some fish have those additional ones. Those all arise from cranioplacoids. Now, in addition, this neural crest is a bundle of cells that then signals different regions for the developing brain. So cranioplacoids become the concentrated bundled sensory system. Neural crest patterns and becomes the different regions of the brain. Okay, and so you can see this, the mesencephalon, rhombencephalon, those all fold over and develop and become the vertebrate brain. So in addition to cranial placodes, which we have sensories, we have the complex brain and nerve connections that allow us to make full use of those sensory systems, right? So now we have three things for vertebrates, right? We've got the notochord becoming the vertebra, we have the cranial placodes becoming those complex sensory systems, and then we have the neural crest, which helps to pattern and become the brain. Okay? Now, number 4 on our list is jaws. And not like the movie, but the movie is named for this feature, right? Really, this was an amazing breakthrough. All of the early vertebrates had a mouth, but had a very limited system of how they could get food because they didn't have complex structures there to help acquire and process that food, and that's what jaws are. Now, jaws are actually modified pharyngeal gill arches. So remember that feature in orange in our early, we'll go back here. Here we go. In orange, in our early vertebrate embryo, those are gill arches. They support and develop into the gills and the gill rakes, these bony or cartilaginous structures that support the gills in the fish. And those were present in the very earliest vertebrate ancestor. Eventually, the first 2 pairs of gill arches become modified for jaws and jaw attachment points. So the jaws are modified gills. Now, the way this works is something that you are familiar with. Nested expression of genes. You don't need to know the names of these if you want the DLX genes, but nested expression of genes that are targeted by these upstream genes to become different things. So DLX 1 through 7 is expressed in the most the most ventral part of these, and that's what patterns these lowest connections. In the medial part, we lose one of those genes. Actually, I think, do we have 3 of them? Yeah, we lose 2 of those genes. We lose 3 and 7. And so all of them are expressed here, only 4 of them are expressed here, and that patterns the lower jaw. And then where only 2 of them are expressed, that patterns the upper jaw. So this kind of thing where they're made all in the same place, but then at different rates, and so some of them can't diffuse as far and only found near where they're made. Others are made at higher rates and so can diffuse much farther. So this one simple signal with various levels of expression and then diffusion of those different levels of expression patterns the jaw. So again, just review and go over that very general. I don't care that you know the name so much. Hox genes are involved in the gill arches, but notice, interestingly enough, the most anterior part of the jaw is actually patterned by a different, closely related, but different set of genes than the Hox genes. Okay? So we have a repression of that gene expression in the anterior end that allows the gill arches to develop differently, and we end up with jaws, and then this nested domain of these DLX genes. You can see that here in a developing vertebrate embryo where they're stained very, very darkly here, and then we get a medium stain, and then finally a lightest stain on the outside in yellow here, and those become the different parts of the jaw. Here it's kind of diagrammed what we're looking at, what we're seeing there. Okay? And notice that we have more than one, so gene duplication played a role here also. These are all very closely related genes that duplicated and allowed for that multiple levels of control from that duplicated gene. Now, the final vertebrate one that we're going to talk about really is just in the tetrapod vertebrates. And we've talked about this a little bit. This is the autopod, or the most distal element of the limbs. So hands and feet. Now, in the earliest fish, all we had was an elongate fin. The first shift was a change that we have, instead of elongate fins along the whole body line, we have 2 regions, and most fish lost the hind region. So if we look at all of the chondriichthyes, all of the osteichthyes, right, these actinopteridae, those are the bony fish, they only have pectoral fins. So they lost what is thought to be the ancestral condition of having both pectoral and pelvic fins. However, the sarcopterygians, which include today's modern lungfish, kept both the forelimbs and the hind limbs. However, they were very simple, right? They had these kind of branching bone-like patterns, but they did not have any real complexity. They're just used as a simple paddle for steering as the fish propels itself through the water. Now, eventually, this proximal to distal axis becomes curved over. So you can see in purple here, this proximal to distal axis becomes curved over. These bones become part of the wrist bone, And these outside projections eventually become articulated and become our digits. Now, the earliest tetrapod vertebrates had all sorts of different combinations of digits. Some of them had 6, as pictured here. Some had 8. I think that go as many as 13 or 14 digits, and everything in between. And so, although there was no set number of digits in the very earliest vertebrates in this autopod, eventually, for whatever reason, we're not sure exactly why, but for whatever reason, 5 becomes the ancestral number. And so all modern vertebrates are descended from an ancestor with just 5 digits. So that diversity gets kind of settled down. Now notice, same old thing, Hox genes here. Not only are they important in patterning the brain, but they also are critically involved in distinguishing what these digits are going to, or what these appendages are going to become. So originally, Hox gene 3, 4, 5, or these are, I think, are supposed to be segments, but these expression areas did the same thing as these expression areas. But after gene duplication, we end up with different downstream targets, and now we can differentiate forelimbs and hind limbs, which many vertebrates do. So here we go. Now, the other key feature to autopods is not only the Hox genes responsible for differentiating forelimbs and hind limbs, they also play a secondary role in the development of the autopod, which is unusual. In insects, for instance, Hox genes then target lots of other genes that then would grow the forelegs or the hind legs or so on. The Hox genes only do kind of the supervisory role. But here the Hox genes play both a supervisory role and they also play a role in the downstream patterning of that. Okay, so there's this extra layer of Hox gene expression in vertebrate limbs and appendages that we don't see in other groups' appendages. All right, those are the 5 big features, right? The autopod, fins to limbs. It's saying example 7 here, but that's from another list. So really we have 5, so let's review them again for the vertebrates. We've got the notochord, number 1. We've got the placodes, number 2, which become these sensory organs. We've got the neural crest, number 3, which becomes the brain. Number 4 are jaws, and we looked in a little bit more detail about the patterning and genes of those jaws. And then finally, number 5, this is just a misprint. In fact, we'll fix it because it bothers me. Example number 5. Fins to limbs. So that's the final example of what made vertebrates so successful. So really many, many key innovations layered upon one another to make this incredibly successful group.